We have seen that our immune system can synthesize millions of different antibodies. These antibodies are synthesized by B lymphocytes and they get inserted in the cell membrane of the B lymphocyte. They are now known as antigen receptors. So when any antigen, and majority of the antigens are present on some pathogen. When any pathogen enters our body for the first time, its antigen will come across some antigen receptor which is complementary to it. So this antigen will bind to the complementary antigen receptor on the surface of a B lymphocyte. Immunization is a process by which we produce active immunity against a pathogen or an infectious agent purposely. Normally, we are exposed to a variety of infectious agents or pathogens. And once we are exposed to this pathogen, a primary response is created. A primary response is produced. The memory of the pathogen is retained. So this is a natural development of immunity against the pathogen or the infectious agents. But in immunization, we purposely produce a primary response before we are exposed, before we are actually exposed to the infectious agents or the pathogen. And this is done normally by giving a vaccine to a healthy person. The process is also known as vaccination. This vaccine contains a small dose of the antigen which is present in the infectious agent or the pathogen. So when we give this small dose of the antigen, it will produce a primary response. There will be a small rise in the antibody concentration against the pathogen, and this response will be short-lived. But some of the B lymphocytes which have encountered this antigen or pathogen will be converted into memory B cells. They will retain the memory of this antigen. And if in future we are naturally exposed to this pathogen, secondary response will develop, and this secondary response will protect us against the infection or the pathogen. So the purpose of giving vaccine is to produce a primary response purposely so that our immune system retains the memory of the antigen or the pathogen. After we have been immunized against a pathogen, if the same pathogen enters our body in future by way of natural infection, a quick and strong secondary response will develop, and this secondary response will either neutralize the pathogen or it can even destroy the pathogen. So we are protected against the natural infection. Immunization is used to prevent a variety of infectious diseases. In fact, the newborns are given a number of vaccines to prevent some common infectious diseases. The vaccine that we give may be made up of killed microorganisms. It may be made up of live attenuated microorganisms. It may contain toxoids, or sometimes a pure antigen is present in the vaccine. When we use killed microorganisms, since the microorganisms are not live, they cannot cause infection, they cannot cause disease. But the antigen is still present in the microorganisms. So this antigen will evoke a primary response, the memory will be retained, and we will be protected against future infections by the same microorganism if it enters our body in live form. Some examples of vaccines that contain killed microorganisms are typhoid vaccine, cholera vaccine, and SARS polio vaccine. In some of the vaccines, we use live attenuated microorganisms. So the microorganism has not been killed, but it has been attenuated. By attenuated, we mean that the microorganism has been made weak, so weak that it cannot cause infection, it cannot cause disease. But the antigen is still present in the microorganism. This antigen will evoke a primary response, and we will be protected against future infection by the same microorganism if it enters our body in a live form. Examples of vaccines that contain live attenuated microorganisms are Sabin's polio vaccine and BCG vaccine against tuberculosis. Some of the microorganisms release toxins, and their harmful effects are caused by the toxins. In such cases, we can use toxoids in the vaccine. A toxoid is prepared by modifying the toxin. The toxin is so modified that it is no longer toxic. It cannot produce toxic effects, but it is still antigenic. So when we give this toxoid, an immune response, a primary immune response will develop, and we will be protected against the pathogen. The examples of vaccines containing toxoids are tetanus toxoid and diphtheria toxoids. Sometimes we give pure antigen to evoke an immune response, a primary immune response. An example is hepatitis B surface antigen. Now, this hepatitis B surface antigen is a protein which is present on the surface of hepatitis B virus and which is responsible for the antigenicity of the virus. Nowadays, this hepatitis B surface antigen is being produced in the laboratory by recombinant DNA technology, and this pure antigen is being used to produce immunity against hepatitis B infection. The vaccines that we give to healthy persons to protect them against various infectious diseases must be effective, and an effective vaccine should be safe, it should be protective, and it should have a sustained effect. By safe, we mean that the vaccine itself should not cause infection or disease. By protective, we mean that once a primary response has developed against the pathogen, if we are exposed to the live pathogen in future, the vaccine must give us strong protection against disease or infection. And as far as possible, the protection should be long lasting. But sometimes the protection is not very long, and then we have to give vaccine at intervals like every year and so on. The immunity which is produced by vaccination is known as active immunity because it's the immune system of the person himself that will defend him against the infectious agents. But this active immunity doesn't develop immediately after vaccination. It takes time. And sometimes there are situations where immunity is required immediately. For example, a person has been exposed to an infectious agent, a dangerous infectious agent. The immunity is required immediately. We can't give a vaccine and then wait for the active immunity to develop because that will take some time. So in such a situation, we can produce passive immunity in the susceptible individual by giving him preformed antibodies. So these antibodies are not his own antibodies. These are antibodies obtained from elsewhere. But once we administer these antibodies, they will give immediate protection against the infectious agent. But the protection will be short-lived. We have seen earlier that the half-lives of immunoglobulins vary from a few days to a few weeks. So the antibodies that we administer will be destroyed after some days. And these antibodies that we use for producing passive immunity may be obtained from animals. An animal has been exposed to a pathogen. The animal has synthesized the antibodies. So we can isolate the antibodies from the animal and we can give these antibodies to the susceptible human being. Or we can obtain these antibodies from some other person who has suffered from the same infection earlier. So his system has already synthesized the antibodies against this particular pathogen or the infectious agent. So we can obtain the antibodies from such a person and give them to a susceptible person. Or we can synthesize the antibodies in laboratory. The technique by which we synthesize antibodies in laboratory is Hybridoma technology. And the antibodies which are synthesized by Hybridoma technology are known as monoclonal antibodies. So these monoclonal antibodies synthesized by Hybridoma technology are being used widely. They are being used for academic purposes. They are being used for diagnostic purposes. Many infectious diseases are diagnosed by performing an antigen antibody reaction. For this reaction, we require pure antibodies. So the antibodies produced by hybridoma technology can be used for diagnosis of infectious diseases. And they can be used for therapeutic purposes to give passive immunity to a susceptible individual. Monoclonal antibodies are clones of each other. They are exactly identical. And as we have seen, they are synthesized in the laboratory by hybridoma technology. In hybridoma technology, we use hybridoma cells to synthesize the antibodies. And a hybridoma cell is a hybrid cell which is prepared by fusing a B lymphocyte with a myeloma cell. As we have seen, the B lymphocytes are antibody forming cells. They synthesize antibodies. And a given B lymphocyte can synthesize only one particular antibody. The myeloma cells are cancer cells. And like other cancer cells, they are immortal cells. All the cancer cells are immortal cells. So myeloma cells are immortal. When we fuse a B lymphocyte with a myeloma cell, a hybridoma cell is formed. And this hybridoma cell possesses some of the properties of myeloma cells and some of the properties of B lymphocytes. Like a myeloma cell, the hybridoma cell is immortal. So it acquires the property of immortality from the myeloma cell. And it synthesizes one particular antibody. This is a property which it has inherited from a B cell. So when we prepare a hybridoma cell by fusing a B cell with a myeloma cell, and we put this hybridoma cell in a culture. This cell will go on dividing forever. It will never die. And it will go on synthesizing the antibody forever. And since only one B lymphocyte has fused with one myeloma cell, the hybridoma cell will synthesize a monoclonal antibody. So all the antibodies will be identical. They will recognize only one particular epitope of one antigen. Now, to prepare a hybridoma cell, we inoculate a mouse with an antigen. So we administer the antigen to the mouse. Then B lymphocytes are taken from the spleen of the mouse, and these B lymphocytes plus the human myeloma cells are put together along with the chemical polyethylene glycol. Now, this polyethylene glycol causes fusion of cell membranes. So when we put different cells together along with polyethylene glycol, the polyethylene glycol will fuse the cell membranes. So when we put B lymphocytes, myeloma cells, and PEG together, some B cells may remain unfused. Some myeloma cells may remain unfused. Some B cells can fuse with other B cells. Some myeloma cells can also fuse with other myeloma cells, but some B cells will fuse with myeloma cells, and the resulting cell will be hybridoma cells. So fusion of B lymphocytes and myeloma cells produces hybridoma cells. But these hybridoma cells are present along with the unfused B lymphocytes and unfused myeloma cells. So we have to separate, we have to select the hybridoma cells from the other cells, the unfused cells, or the B cells which are fused with B cells, the myeloma cells which are fused with myeloma cells. And we can select the hybridoma cells by culturing all the cells, by putting all the cells in a culture medium, which is known as hypoxanthine aminopterin thymidine culture medium, or HAT medium. So we inject a pure antigen into a mouse, we take out its spleen cells, we take some myeloma cells, and we put these myeloma cells, the spleen cells, plus polyethylene glycol together. So some of the B lymphocytes will fuse with some of the myeloma cells, and hybridoma cells will. And by putting all these cells in HAT medium, we select the hybridoma cells. Now, why do only hybridoma cells survive in HAT medium, and why not the unfused B lymphocytes and unfused myeloma cells? The myeloma cells do not possess HGPRT, hypoxanthine guanine phosphoribosyltransferase transferase. This is a salvage enzyme that can convert hypoxanthine into inosine monophosphate. And from inosine monophosphate, adenosine monophosphate can be synthesized, and guanosine monophosphate can be synthesized. Now, we have put hypoxanthine in the culture medium, that is HAT medium, but because of absence of HGPRT, myeloma cells cannot convert hypoxanthine into IMP, and therefore they cannot form AMP and GMP. So the myeloma cells, the normal myeloma cells, are dependent only on de novo synthesis for their requirement of purine nucleotides. But we have added aminopterin to the culture medium. Aminopterin is a competitive inhibitor of dihydrofolate reductase. So in the presence of this inhibitor, dihydrofolate will not be converted into tetrahydrofolate, and in the absence of tetrahydrofolate, de novo synthesis cannot take place. So myeloma cells cannot obtain purine nucleotides from salvage pathway, and they cannot obtain purine nucleotides from de novo synthesis. The unfused B lymphocytes or the B lymphocytes which have fused with other B lymphocytes will die soon when their lifespan is over. The unfused myeloma cells cannot survive because purines are not available. They have been deprived of purines. So in HAT medium, only the hybridoma cells will survive because they have obtained HGPRT from B lymphocytes. So in HAT medium, this de novo synthesis of IMP is blocked by aminopterin. The salvage of hypoxanthine is blocked by absence of HGPRT. The synthesis of TMP from UMP, there is synthesis of thymidine monophosphate from iodine monophosphate, is also blocked by aminopterin. But the hybridoma cells can salvage hypoxanthine. They can synthesize IMP, and from IMP, AMP can be synthesized and GMP can be synthesized. Moreover, these cells possess thymidine kinase, and we have added thymidine in the culture medium. So this thymidine can be converted into thymidine monophosphate by the hybridoma cells. So hybridoma cells can form purines as well as pyrimidines, and therefore only these cells will survive in the HA. We will continue this discussion in part 4. Thank you.